Hello, everyone, and welcome to today's webinar on an introduction to population enrichment designs. This webinar is part of our new introductory webinar series on complex innovative trial designs. I am pleased to introduce our presenter today. Dr. Thomas Burnett is a Senior Research Associate in Medical and Pharmaceutical Statistics at Lancaster University, where his main research interests are adaptive clinical trials and personalized medicine. He studied for his bachelor's in mathematics and statistics with placement at the University of Bath, spending a year working on sample design at the Office for National Statistics. Thomas holds a PhD from the University of Bath, where he had a case studentship that involved working closely with Roche products, researching the Bayesian optimization of adaptive enrichment trials. It is now my pleasure to turn it over to Dr. Thomas Burnett.
Okay, thank you, Alyssa. Hello, everyone. I'm now going to just quickly turn my webcam off so that we can focus on the slides instead of me as we go. So firstly, thank you to Cytel for inviting me to speak at this event and for the close collaboration in terms of putting the slides together. And hopefully we can have a successful event and pass on a little information on adaptive enrichment. So in terms of today's talk, just to give you a little idea of what to expect, we'll give you kind of a brief introduction to what population enrichment is and where it fits in terms of as a clinical trial. Then we'll talk about a couple of the crucial aspects in terms of how adaptive enrichment or population enrichment designs come together. So the hypothesis testing in order to make formal analysis, and then the decision making, which is kind of a key component of many adaptive designs. We'll talk through a few different methods that we could approach this with. We'll then go through an example of adaptive enrichment in the TAPAS trial, just to show you a real-world example of where it was used. And then we'll hand back to Alyssa, who will go through essentially the construction of the TAPAS trial, but from EAST, and talk to you a little bit about the ENRICH module in particular in EAST. Okay. So what is population enrichment? I'm going to go from very basic principles and we'll slowly build our way there. So to start off with, for any clinical trial where you're developing a new treatment, we're all familiar with the idea that you may have a target population where you expect to use the treatment. So people with the thing that you're trying to treat, or maybe a subset of those people who have certain patient characteristics that you're interested in for the trial. And within that classical setting, we would think about maybe a controlled clinical trial in which we randomize patients both to an experimental treatment and to a control treatment. And this allows us to test the null hypothesis that there is no difference between these two treatments. So in this case, we're looking to reject the null hypothesis and say that the experimental treatment provides some benefit to patients over the control. So what about this population? Just a fact that we may have kind of in the background is that we may have subgroups. So we may expect for these subgroups to behave or interact differently with the treatment that we're applying. So we have a very simple example here. We have the blue people and the yellow people who we split out into population 1 and population 2. So that within these two populations, so as I was saying, within our target population for the treatment, we may identify these patient subgroups that we expect to interact differently. Now, the key point that we're going to make use of for adaptive enrichment is that these subgroups or subpopulations, as we'll refer to them, are predefined. That is, we know of their existence before the trial begins, and so we're going to make use of these known subgroups in order to conduct our trial. So what does this mean in terms of the study? Well, there are a few different ways that we can define our null hypotheses. So previously, when we talked about the target population, we talked about the null hypothesis being the difference between the experimental, an examination of the difference between the experimental treatment and the control. And now we can consider making that examination but within each population. So making that examination within population 1, making that examination within population 2, for which we define our H01 or our H02, just scripting based on which population we're looking at. We could alternatively consider testing in the full population, so the combination of the two subgroups. Again, this would just be a comparison of experimental treatment and control. But I think for today, we'll mostly focus on the case where we're looking really at population 1 and population 2. Okay, so how could we study this treatment in a classical setting? So by classical setting, I mean a non-adaptive design. That is, the sampling for the design is set at the start of the trial. You run all the way through the trial, and then at the end of that sampling, you have your analysis. Well, we could conduct the trial in just a single population. So in this case, in population 1, using the same amount of resource, i.e. the same number of patients, we could conduct the trial in population 2. Again, this is an examination where we'll compare the experimental treatment and control. Important to note, by splitting into experimental treatment and control here, what I'm really referring to is randomization. So patients being randomized to the experimental treatment and the control treatment. For today, we're going to assume that randomization is handled in the background, but in some suitable way as we request it. So the next alternative is to consider the full population. So that is recruiting everybody into the trial. We could make that test just in the full population. So as we saw on the previous slide, H03 refers to just testing the difference between the experimental treatment and the control treatment in that full population. Or alternatively, we could recruit everybody and then test within the subgroups. Okay, one thing you might notice here compared to the previous couple of slides is the number of patients. If we take that as fixed, we have less patients within each subgroup or subpopulation when compared to conducting the trial in that population only. So we're assuming maybe some finite resource. That, or you could consider that you might increase the sample size of the trial as a whole in order to recruit the same number of patients to each group. So the question for us at this point in a classical setting is, well, how do we choose which populations of patients to recruit and therefore study in the trial? So imagine a situation where you have different members of the trial team with different opinions. Some are saying that, well, population 1 is the group that we should really study. Others are saying, well, it's population 2. And others are saying, well, maybe we should just recruit both. You can see that what we would really like to do is compromise between the options that we've seen previously to really reflect our uncertainty about which patients are receiving the benefit. So this is exactly what we do in a population enrichment design. We begin by recruiting every patient possible into the trial for some portion of the total sample size. So illustrated here, we have perhaps half of our total sample where we're recruiting from the full population and again randomizing those patients to the experimental treatment and to the control treatment. At this stage, we then make a choice about how we recruit the remainder of the trial. So we have half of our patients remaining, and what we will do is choose from these 3 options based on the observations from stage 1 of the trial. So in stage 2, we recruit either in population 1, if population 1 looked promising in the first stage of the trial, in population 2, if population 2 looked promising in the first stage of the trial, or continue in the full population if we think that that is the most promising approach for the remainder of the trial. Worth noting that again, patients or participants will continue to be randomized in the second stage of the trial. So we still have that comparison of experimental treatment and control within each stage of the trial. So just a few considerations about how we might make this choice. Essentially, we need data to be available such that we're making an informed decision at the interim analysis. So that is data that is perhaps quickly available. In that case, there's not a long delay between the recruitment of a patient and the observation of their primary endpoint. So we can make an informed choice based on that patient quite quickly. But now imagine you're in the case of a delayed response. In this setting, you may have some delay between the recruitment of the patient and when you observe their endpoint. This causes a slight problem in an adaptive trial in that you can't affect the recruitment of that patient or the randomization of that patient anymore, depending on which type of adaptation you're looking at. But you also don't have the benefit of the information from that patient in order to inform your interim decision making. So this is a problem that is also seen in survival data. You have patients who have been recruited to the trial who are yet to have any information from in terms of making your interim decision. There are a couple of ways that you can think of to get some of that information back. So imagine you have repeated measures on a patient. Imagine the primary endpoint is measured after 6 months, but you take a similar measure after 1 month, where you can use those repeated measures to gain more information about where that patient might go. You could consider, instead of overall survival, progression-free survival, or you could consider surrogate endpoints. And then the important thing that this allows us to highlight is that there is a trade-off in order to achieve the flexibility. So in this case, we're seeing that trade-off in terms of patients who have been recruited but not observed. Essentially, we have no influence over what happens with those patients, but we also have no benefit from understanding what their observations will look like. So as a quick summary to this introduction to what adaptive enrichment is and what it's trying to achieve, we're working with predefined subgroups. We have pre-planned adaptation options. So in the example that we've seen pre-planned options of continuing in one population, continuing in the other, or continuing in both. This allows us to offer a compromise between the fixed sampling alternatives where we would have had to make that decision before the trial began. Instead, we're able to make it based on data-driven decision making at the interim analysis. We can apply these methods in multiple data types and multiple settings. But the main key point is that it's desirable that we can make a well-informed decision at the interim analysis. So we're going to take a moment to pause here and ask if you have any questions about this kind of broad introduction that we've gone through so far to adaptive enrichment or population enrichment before we get into some of the more nuanced issues. I believe we have Alyssa who will be kind of running the questions for us at this stage.
Thanks, Tom. And as a reminder, you can submit your questions to the questions pane in your attendee control panel. So we have one question here. Can you extend your design to more than 2 groups?
Good question. So obviously within the presentation as we've seen it, we've only considered 2 subgroups. And you can easily think of examples where you might have more subgroups. And of course, theoretically, this is possible. Typically within the literature, you see application to 2 subgroups, but with commentary on how to extend the methods. So it's certainly something that you could consider in the case of a real trial. Yeah.
Thanks, Tom. And let's see, one last question here. How do you make a choice of which population you would like to enrich?
It's a great question, and it's a question we'll actually come back to later in the talk. So, as I said, this will be based on interim observations, so observations available from patients recruited in the first stage of the trial. And based on these observations, there are a few different ways to go about that choice, and we'll see those as we continue forward.
Great, thanks, Tom.
Okay, so at this point, we'll continue moving through, and we'll start talking about hypothesis testing. So for those of you that are very interested in the statistics of what's going on here, you may already be concerned that we have a couple of key issues that might be going on in terms of making formal inference. So we have multiple groups and which causes multiple null hypotheses. And we also have a multi-stage trial. So we need to do a little bit of work in order to consider how to handle this. So as a recap, we have potentially null hypotheses in each population or in the combined full population, and we study those over multiple stages. So what does this multiple testing problem mean? Well, in a typical single hypothesis trial, you would be concerned with falsely declaring efficacy. That is making a type 1 error, which is to say you would reject the null hypothesis that there's no difference between the treatment and control when it's in fact true. In the multiple hypothesis setting, we're still concerned with that same essential problem of falsely declaring efficacy. But because we have multiple arms, we restate the error in a slightly different way. So a family-wise error in this setting would be to reject one or more true null hypotheses. So that is, within one or more subgroups, we declare that the experimental treatment is significantly different from the control, when in fact it isn't. In particular, in a confirmatory trial setting, we would be particularly concerned with controlling the error rate at some pre-specified level. And so this is known as strong control of the family-wise error rate in the multiple hypothesis setting. And that's simply the probability of rejecting one or more true null hypotheses is controlled at some predetermined level that we'll note down by alpha in this case. So how do we ensure we achieve strong control? Well, a critical fact that we're going to lean on is that we have pre-planned trial options. So as we said before, we can either continue recruitment in population 1, continue recruitment in population 2, or continue recruitment in both. If we continue recruitment in population 1, we test the corresponding null. 2, we can again test the corresponding null. Or if we continue in both, we may be interested in both. And again, you could add in a hypothesis for the full population in this third case, where you continue recruiting both groups. So what we need is a testing procedure that will cover each of these possible eventualities of the trial in terms of controlling the error rate. What we require is a closed testing procedure. So these first two tests should be familiar in that they are the individual tests of the null hypotheses in population 1 and in population 2. The addition is this intersection hypothesis. And I do apologize for those that aren't particularly fond of the statistical side. We're going to get a little bit technical for a couple of slides here in terms of how to test this, but we will come back to out of that technical setting quite quickly. So please just bear with me. It will all make sense again afterwards if you don't fully follow. So this intersection hypothesis is slightly new, but all it means is that both null hypotheses are true at the same time. So in order to globally reject the null hypothesis, that is, at a trial level, we can say that we've rejected the corresponding null. Well, in population 1, this happens when we reject all tests involving population 1, so that individual level alpha test that is constructed as normal, and a test of the intersection hypothesis. Similarly, in population 2, it's all tests involving population 2 that we are concerned with, and so that individual local test and the intersection hypothesis again. The big question is, how do we test that intersection hypothesis? Well, there are many ways, but we'll take a quick example, a quick detour through Symes' method. And so imagine you have p-values for testing the individual hypotheses. To test the combined hypothesis, you simply apply this function. So it's the minimum of 2 times the minimum of the p-values or the maximum. What this essentially means is that you either reject both null hypotheses at level alpha, or you reject one null hypothesis at level alpha over 2. And so this is another of those points where you're paying some small price for the adaptive nature and for the multiple testing. In addition to the multiple hypotheses, I also stated that we would have multiple stages in the trial, and that's something that we need to deal with. Well, this is just one simple method. So from our comparison of the experimental treatment and the control treatment within each stage of the trial, we might find our corresponding test statistic. So a Z statistic that's, say, normally distributed. We can then find a combined test statistic simply by weighting these two things together. Worth noting that these weights need to be predefined before the trial begins in order to control the error rate in the desired way. This particular method is known as the weighted inverse normal, but again, you can consider other possible methods. So overall, how do we put these two things together? Well, within each stage, we might apply Symes' method to get our intersection p-value from our individual p-values. So we see that in both stage 1 and in stage 2. And then we combine everything together using a combination test of the form seen on the previous slide, such as the weighted inverse normal, to test each hypothesis globally. And so you're just combining the p-value from the first stage and the p-value from the second stage within the subgroup that you're currently testing. Okay, that's it for the more technical part of the talk, but just to run through what are the key points in terms of the things that you need to consider. So we have predefined pathways through the trial, and this is critical because it gives us a fixed list of possible hypotheses that we might be interested in testing. This allows us to construct a test procedure that controls the error rate for all possible decisions, which is critical for what we'll go on to do next. Because what this means is that, as is written in bold here, the error rate is always controlled no matter what decision we might make at the interim analysis. This means that we have a lot of freedom in terms of how we make that decision, and that's what we'll go on to talk about next. So with hypothesis testing in place, let us have a little bit of a think about how you might go about making decisions. As a recap, we're going to make these decisions based on the available data or the available information from the first stage of the trial. Remember that in that first stage of the trial, we've recruited from all populations and we've randomized patients to the experimental treatment and the control treatment to allow us to make a comparison between the two. So what we get from this are estimates of the treatment effect. So in population 1, we have our first stage treatment effect estimate. And in population 2, again, we have our first stage treatment effect estimate. And it's these estimates that we'll base our decision making on. Of course, you could incorporate more information from the first stage of the trial into your decision making process if you felt it was necessary. And we'll talk about a couple of ways that that might come into play, kind of implicitly within a couple of the methods that we look at. But as I said, you have complete freedom as to how you make this decision. And so if you wanted to make more full use of this information, or even information external to the trial, you could. So let's start off by thinking about a really simple decision rule. And that is that we simply select the best population at the interim analysis. Well, this is quite easy to apply in that we simply look at the treatment effect estimates from the first stage of the trial, we compare them, and we select the appropriate subpopulation based on what we've observed in the first stage. Obviously, this is appealing because we then take forward the population within which we think that the treatment really does work, and that's clearly desirable. Perhaps a little simple though. Perhaps we might want to consider something slightly more complex, such as threshold selection. So in this case, we say if the treatment effect is beyond a certain threshold value, then we continue recruiting patients for that subpopulation. So this threshold we may set to some clinically relevant point where we say we would like the estimate to be over this value if it's demonstrating that there is actually a meaningful difference between experimental treatment and control. Or we could tune these thresholds to hit certain operating characteristics of the trial, depending on what it is that we want to achieve. The important thing is, within this type of setting, this would allow us to look at continuing to recruit in one subpopulation, the other, or continue recruiting in both if both are showing sufficient benefit to the patients within the trial. So I'm going to take a little detour from the kind of more classical statistical setting here and talk to you about something quite close to my heart. So Bayesian decision making and adaptive enrichment was the topic of my PhD thesis and is an alternative way to consider how you might make decisions at the interim analysis. So the crucial difference in Bayesian statistics is the capturing of prior uncertainty about the treatment effect before the trial. So for those of you that aren't familiar, this prior uncertainty may consist of expert opinion, pre-existing information on the treatment, a combination of the two, and prior elicitation in itself is a big exercise to talk about, and it's something we'll kind of skip over here. But the point is we have some kind of other source of information that we wish to incorporate in the decision making. Crucial to say that the prior information that we use here doesn't contribute to the hypothesis testing. So as I said, the hypothesis testing is set up to give us complete freedom in terms of how we make interim decisions. That means that we can use this Bayesian decision making without impacting the hypothesis testing of the trial. We're purely using the Bayesian methodology for decision making at the interim analysis. Okay, but at the interim analysis, we now have some observations from the trial that are giving us some estimates of the treatment effect after the first stage. We can combine this with our prior distribution to give us some kind of posterior. And instead of it just being the estimates of the treatment effect that inform our interim decision, it's now our posterior distribution that will inform our decision at the interim analysis. So just a quick note as to why we might want to do this and what it might look like. We can imagine what a prior distribution and a posterior distribution might look like in kind of an idealized case. And so the black curve here represents the prior distribution. So this is our beliefs about what the true treatment effect theta is. And we can see that it's quite dispersed. Hopefully, at the interim analysis, based on our interim data, we'll be able to update this prior distribution to our posterior, where you can see that we are covering less of a range of values over the center of the distribution. So we have hopefully some more certainty based on the trial observations about which population we should be giving the treatment to. Now this comes back to what I was saying right at the start of the presentation, where we talked about we have this uncertainty about which groups we should conduct the trial in. Essentially what we're doing here is capturing that uncertainty in some statistical distribution. Now if you did have a tight prior distribution with high certainty before the trial began, then actually you might not consider a population enrichment design because you may already be confident enough to make a selection of the population before the trial begins. So just another useful tool that comes out of this framework is defining a single measure of overall trial performance. So you'll note that we, within the two hypothesis settings, so one in population 1 and one in population 2, we essentially have two outcomes of interest. One is to reject the null hypothesis in population 1. The other is to reject the null hypothesis in population 2. What a gain function or a utility function does is bring those possible outcomes of the trial together and weights them by some kind of meaningful benefit in order to reflect how useful actually is this result to patients or to the sponsor. So here, your choice of A and B will be chosen to reflect the benefit that the corresponding result of the trial brings. So for example, you might consider from a patient perspective, there's more benefit to a true higher treatment effect. Also beneficial to the sponsor to be able to treat patients in a good way. To a sponsor, it may also be important to have a high estimate of the treatment effect because this will also assist with demonstrating the effect of the treatment and marketing the treatment and so on and so forth. But the key point is that this gives us a single measure of performance based on the possible outcomes of interest. So how do we use this utility function and this Bayesian framework to make decisions? Well, our aim is to maximize the expected value given the present understanding of the treatment effect. So in our population enrichment example, this means that after stage 1, we have our 3 possible choices, and we wish to maximize what we expect the gain function, the value of the gain function to be, given our current understanding, so our posterior distribution in this case. This requires some complex simulation that we won't go into here, but essentially it's fairly simple from an implementation standpoint once in the actual trial, because you simply evaluate which looks like the best option given my utility function and my current understanding of the data to choose going forward with the trial. Okay, stepping back away from the Bayesian setting, and now we'll look at the promising zone approach. And it may be a little confusing to jump back and forth between the 2 methods, but we're going to use this promising zone approach in the example to come. So I thought it was most useful to discuss this at this point. So the idea of the promising zone approach, we're going to look at a 3-option version at the interim analysis. So here we're assuming that there's one population which we think is probably favorable for the treatment or receives a higher benefit for the treatment. And the idea is that we will either enrich into that population, so continue recruiting, but only from that subpopulation. We might be in a favorable zone where we continue the trial as it is, or we may be in a promising zone where we might actually choose to recruit more patients. And so in this promising zone, we also are able to kind of review our sample size for the trial moving forward. The important point of how this is constructed is that this choice is made based on the conditional probability. So again, given our observations at the interim analysis, what is the probability that we now reject the null hypothesis? And this is the approach that is implemented in the ENRICH package in East that will be spoken about later. Worth noting that obviously this isn't the only way that we might set this up. So we may also consider an unfavorable zone, or particularly useful, something we haven't spoken about in any of the examples so far, is a futility zone. So this might be where no subgroup actually looks to be benefiting from the experimental treatment. And so we might be in a futility zone where we choose to stop the trial early, i.e. to protect patients from a treatment that we have no evidence that is actually working. Okay, so to end my component for now, we're going to very quickly talk through the TAPAS trial that makes use of this promising zone approach. And so this is an example of a real trial. I believe the references for this will be in the notes, as well as references for some of the other sections, should you be interested, when these slides are made available afterwards. So a bit of background about this study. This was a study of TRC105 in patients with angiosarcoma. The important point here was that we were able to identify 2 subgroups before the trial began. So these are the cutaneous subgroup and the non-cutaneous subgroup. There was an indication in this case of greater tumor sensitivity in the cutaneous subgroup. So hopefully, given everything we've talked about so far, this now starts to look familiar and the exact kind of setting that we would wish to apply an adaptive enrichment design within. So what was the design itself? Well, as we talked about earlier, adaptive enrichment can be applied within a survival setting. In this case, the primary endpoint was progression-free survival. Again, we talked about how that may be slightly more quickly available than, for example, overall survival, and therefore might allow you to make more informed decisions at an interim analysis. The initial sample size was 124 patients to be followed until there were 95 events of progression or death. I must apologise because I've not put the figure in here, but I'm sure we'll talk about it later on when you see this example being constructed in East. The interim analysis was obviously part way through this trial, and I'll make sure that this number is updated when the slides are circulated. So what were the possible interim decisions? Well, within this study, the promising zone approach was indeed used. And that defined three possible zones. So there was the favourable zone, in which the study would continue as planned from the outset. There was the promising zone, in which the sample size would be increased, but patients would continue to be recruited from the full population. And there was the enrichment zone, where recruitment only continued in the cutaneous subgroup, and actually there was a slight increase in sample size in this enrichment zone as well, I believe. Worth noting that in this particular trial, an unfavourable zone was also defined, but in this case, the same action was taken as was for the favourable zone, and so we had the same possible outcome in both ways. What actually happened in the trial? Well, in total, it recruited 128 patients. It continued in the full population as planned, so falling within this favourable zone category. And unfortunately, in this case, it did not demonstrate activity. The experimental treatment did not demonstrate activity. What this kind of highlights for us, though, is that an adaptive design is not necessarily a virtue in itself. The flexibility was useful in this case, as had we found a particularly strong effect within this subgroup, we may have been able to usefully focus our attention there. But that doesn't ultimately change what the overall outcome of the trial may be. You still, in this case, this would look identical to a trial that recruited from the full population throughout. And so we have the same data. Okay, so at this juncture, I'm going to pass over to Alyssa, who will talk us a little bit through the enrichment module in EAST.
So this is just a reminder that EAST gives you access to a wide selection of fixed and adaptive trial designs, and you can use EAST to help you quickly create and compare those trial designs. So one of the modules that we have in EAST is the ENRICH module, and I'll just briefly show you how you can reproduce the TAPAS trial using this module. And so we have a few tabs where we enter the relevant parameters for the trial before we simulate on that design. And so what I'm showing here is a screenshot of the Design Parameters tab, and that's where we enter the event rates and enrollment rates for this survival design. And any other relevant design parameters like accrual rate and subpopulation prevalence are entered here as well. And then in the Adaptation Parameters tab, we can specify the parameters for both the promising zone and the enrichment zone. And this includes specifying the maximum sample size and number of events if we were to adapt, as well as the lower and upper bounds for the promising zone and conditional power for entry into the enrichment zone. And so here's an example of a graphic that you will find in the ENRICH module. This graph here allows you to visually see the number of subjects and events over time. And if you see in the right-hand corner here, you can examine each cohort separately or both cohorts combined. And lastly, here's an example of the results that are generated from this simulation. So you'll see this first table here shows the zone-wise summaries of the simulation. And then we also have a summary of the overall simulation results and the results for just the simulations that fell into the enrichment zone. So this was just meant to give you a very brief overview of the ENRICH module, some of the features that we have available within this module. If you are interested in learning more about ENRICH, we will be sending a follow-up email with a link to a more detailed webinar on ENRICH following this webinar today. So Tom, I'm going to turn it back to you.
Thank you very much. So I think probably just worth mentioning, just before I move on from this slide, I did have a little bit of a play with the ENRICH module myself in the build up to this presentation. And it is quite impressive how quickly some of these simulations run. Okay, so let's just have a little bit of a talk through some of the key points that we've gone through today. And then we'll open everything back up to questions and we can have some more discussion about population enrichment. You'll notice in this presentation I've interchangeably used the terms adaptive enrichment and population enrichment, both meaning the same thing. So the key point of adaptive enrichment is that it introduces flexibility to subgroup selection. So that flexibility being that we're basing that decision making about which population to select for the trial upon trial observations. And so the idea being that we're able to more accurately choose the population or the subgroup who will benefit most from the treatment under study. This flexibility comes at some cost, but we can account for it in terms of our hypothesis testing, allowing us to make formal analysis of what's going on in the trial. And a key aspect of how the trial will perform is the decision making at the interim analysis. So we've covered a few different ways of making those decisions. What we haven't necessarily talked about too much is in terms of actually designing the trial, how do you choose which decision making method to use? So within the select the best, there's not much tuning that can be done, but within the thresholds option that we saw, you can tune those thresholds to make the operating characteristics of the trial behave how you would like. Within the promising zone approach, again, that you can tune the thresholds, but now in terms of conditional probability, to see how often you fall within particular zones. Under the Bayesian approach, we have a slightly different setup in that we are optimal for the gain function that's supplied. But this gain function itself and the prior distribution both require careful definition to really capture what it is that you're aiming to achieve from the trial. And so these several methods that are available, you may find that one is more suitable to allow you to tune the trial to be the way that you want it to be. I wanted to end on the note that used appropriately, adaptive enrichment or population enrichment can be a powerful tool, ensuring that the right patients are getting the treatment that's right for them, a step on the way to personalized medicine, but with a note of caution that it's not necessarily always the best choice. So flexibility of the design is not necessarily a virtue in itself if there are other designs that are fixed sampling, for example, that you are already able to make a correct decision to use. So let's think of a simple example. Imagine in the examples we've looked at today, population 1, you know before the trial it's clearly the better treatment. Well, in that case, I would argue that you may be better off recruiting only from population 1. Adaptive enrichment is useful in those edge cases where you have uncertainty about which populations should be recruited from during the trial. And it's there that it can make a difference, allowing you to receive some small operational benefits whilst also compromising between the other designs that you might consider using. So I believe this is back to Alyssa for a moment to talk a little further about products and services from Cytel.
Thank you, Tom. And this is just a reminder that in addition to software, we offer a range of services from staff that act as an extension of your team and consulting services for more cutting edge projects to our new real world analytics capabilities. We're here to help support your objectives. Okay,
so at this point, I've presented to you the core material that I was hoping to present to you today. So we have now time for some questions. If you have further questions that you don't get to ask in the chat, I'm sure I can be available via email afterwards as well to discuss any aspects that you might be interested in. But for now, we'll hand over to any questions within the chat.
Thanks, Tom. And as a reminder, you can submit your questions through the questions pane in your attendee control panel. So Tom, our first question. How does enrichment impact the generalizability of your conclusions?
It's a good question. So in terms of the generalizability, it will depend on exactly what enrichment decision is made. So essentially, I would argue that your generalizability will be the same as if you had conducted a fixed sample trial or a fixed design, kind of a traditional trial, in the population that you end up looking at by the end of the adaptive enrichment or the population enrichment trial. And so the answer is, if you were to conduct a trial in population 1, the generalizability of the adaptive enrichment trial that specializes into population 1 by the end of the trial is broadly the same. So really think of the fixed sampling counterpart that you end up being most similar to in terms of equivalence.
Thank you. Our next question, how do you choose weights for the stage combining?
It's a great question. So within that particular example that we've looked at, the weights for the weighted inverse normal are optimal when they're chosen to be in proportion to the sample size each side of the interim analysis. So if you have half of your sample before and half of your sample after, your weights would be equal, for example. Now in adaptive enrichment, it's not quite that simple because obviously you have different possibilities in terms of what might actually be chosen at the interim analysis, and therefore how much of the sample size is available within each population before and after this interim analysis. Typically what's done is you make a selection based on what you think is a likely decision at the interim analysis in order to optimize the weights for that likely decision, knowing that you won't be too far off optimal for the other one.
Thank you, Tom. And we have just a couple more questions here. Can you design an enrichment trial with more than one interim analysis?
Theoretically, yeah, it's certainly possible. So all of the material that we've seen here, we've seen everything can be extended to both more interim analyses and more subpopulations. The interesting thing within that setting would be that, let's say you had a threshold approach for argument's sake, you would have two sets of thresholds. So you'd have a set of thresholds for the first interim analysis, and then you'd have a set of thresholds for the second interim analysis. Now, just kind of speaking hypothetically, I haven't looked into actually optimizing that, but I think recently there was a webinar on group sequential designs by Christopher Jennison within this series. You might consider something like that. So within group sequential designs, you see at the first analysis, the thresholds are wider, and at the second analysis, you see the thresholds tightening together. You might wish to consider doing something like that because of your higher uncertainty, the less data you have earlier on in the trial.
Great, and I think we have time here for one more question. How do we calculate the overall effect size with confidence intervals?
So in terms of computing confidence intervals in a multi-stage design, especially with the multiple testing, that's a complex issue and probably beyond what we have time to answer in a quick question. As you've clearly picked up on, it's not a trivial thing to do, particularly in terms of making sure that your confidence intervals match your trial outcome. But it's something that if you're interested in, I suggest maybe we could talk afterwards and I can share some more papers and background material to assist with that kind of work.
Thanks, Tom. And we actually have just one follow-up question to the question about choosing weights. Is it possible to predefine several weight sets?
So in terms of the testing, your weight sets would be defined up front before the trial begins. And so by predefining several, I would assume you might mean predefine them and then choose them based on which decision you make. Unfortunately, that wouldn't be possible because at that point, your testing methodology is becoming dependent on the data from the trial, which is obviously a problematic assumption. So you do need to define precisely what set of weights will be used at the start of the trial. Unless you mean in terms of a different way, could you define different weights for different populations? As long as they're predefined before the trial begins, then yes, I think in principle that would be okay.
Great, thank you, Tom. So before we conclude for today, I am excited to announce our next webinar in this complex innovative design series, and that's happening on August 5th. And it will be on adaptive designs for optimal dose finding, and our presenter will be Jose Pinero. And you'll be receiving more communication about this, this upcoming webinar in the coming weeks. So with that, thank you, Tom, and thank you to everyone for attending today's webinar on population enrichment designs. On behalf of Cytel, thank you for joining us today, and I hope that you have a great rest of your day.
And thank you again to Cytel for the opportunity. opportunity to present and the close collaboration in terms of bringing the slides together.